Alessandro Gozzi.
Okay, thank you, Adriana, for having me here. Good morning. I would like, with my talk, to close this session by illustrating how we can somewhat, try and translate some of the approaches we've been discussing here in this session and other session in this conference onto model organisms such as rodents. And the idea here is basically, to exploit, to leverage, the power that you have in these, rodent models, all the tools that you can use. I'm going to show you some examples to try to advance our understanding of autism connectopathy. So, I'm going to start very briefly by refreshing a key concept that is being, said over and over, which is the fact that functional connectivity is almost invariably disrupted in autism. However, as you probably know, the original expression, the location of these changes and their sign is very heterogeneous across samples, across sites, and so on and so forth. So we have evidence of reduced connectivity, we have evidence of increased, and lately there's also this idea, this emerging of a possible composite, mosaic patterns of hypo. And so, as a result of these discrepancies, we are left, with a number of major key questions in the field as to the origin and as to the significance of these alterations. And there are many of these questions. I'm just listing a few here, which are relevant for my presentation, and so that's why I'm just very briefly show you what some of these relevant questions that we also try to address with the approach, with the paradigm I'm going to show you. And the first is whether and how genetic or the disparate etiologies that have been associated with the spectrum could somewhat contribute to the neuroconnectional heterogeneity that we've been seeing in autism. A second key question is perhaps a little more ambitious. Can we leverage, in fact, this heterogeneity to perhaps identify subtypes of this disorder? This is, of course, a key unmet need in the field. And last but not least, that the brain is a dynamic system, and so there is still a question out there whether we can somewhat relate alterations, if any, in neural dynamics to these static patterns of functional connectivity. And what I'm arguing here is that if you want to understand and unravel a complex and multifactorial phenomenon such as functional coupling, functional connectivity, we must necessarily complement clinical efforts with novel research paradigms in which we can causally link etiological factors with connectivity fingerprints. And so one of such approaches is going to be described by myself in this talk, and this is pretty much an extension of what Clara beautifully showed in her talk before. So she showed that a powerful approach in humans is to select genetically or etiologically homogeneous populations, and you can link these etiologies to some fingerprints, connectivity we've seen so far. This is very powerful, but you all know here in the room that these mutations are very rare, so it is difficult to generalize this approach. So what we propose very simply is to shift this paradigm onto model organisms such as rodents. And by doing that, we can achieve two major advantages. The first one is that by leveraging genetics, for example, or the tools we have, we can probe a limitless number of mutations or etiologies, okay, in very much controlled conditions. You can control environment, so on and so forth. That's a second important aspect here related to this approach. So to speak, it's a bonus feature, which is the fact that in the process we can also try and understand what mechanisms are leading to this functional decoupling or aberrancies. And if we do this by using a translatable readout like resting state fMRI, we have a third important advantage. We can try and translate directly from the clinic and vice versa to the clinic changes across species. And this notion is supported by quite a few studies that we carried out in the lab and that have been, in the meantime, are also replicated by many other groups in the world, suggesting that overall there's somewhat a comparable functional organization in resting state networks between human and mouse. And these fundamental features, for example, are the fact that these networks we see across species are homotopic, they are functionally meaningful, and we also have important integrative networks that are somewhat evolutionary conserves, such as salience like networking rodents, a default mode like networking rodents, and these are, of course, two polymodal networks that are very important and key for autism. And this notion has been further corroborated by a study that we published last year that would support the idea that autism-associated etiologies comparably affect or impair connectivity across species. So how did we come to this conclusion? Well, here we have again the 16p11 copy number validation. This is the deletion. We got the data from the Simons Foundation, and what we did, we wanted to compare the effect of these mutations in human and mouse, okay? And so what we found here, I don't know whether I can point it somewhat, but anyway, yeah, here it is. So what we found here is that there's basically in carriers of this deletion, there's a focal reduction in connectivity in prefrontal areas. This seems to be clinically relevant somewhat because it is inversely correlated with social responsiveness. And we also show that functional connectivity in prefrontal cortex is somewhat desynchronized with long-range areas in parietal posterior regions. So what about the mouse? Well, actually, when we recapitulate this mutation in the mouse, what we get is that pretty much, it's the same picture. We have a reduction, focal reduction in functional connectivity in prefrontal areas, and if you want to see where this long-range disconnection here is, it is affecting again parietal association region. So far so good, very encouraging, right? And this is suggesting that there is, benefit. There is a benefit. There is scope for trying to apply this approach in trying to understand what's going on with this disconnectivity in autism. So, all right. So let me just get back to our original questions. This is a question that's been there for a long time. So, do different etiologies contribute to the heterogeneity we've been seeing? So a very simple way to address this question is by using the approach I show you and somewhat map the connectional landscape, which means, in other words, to apply like functional connectivity measurements across different models of autism. So mouse lines that recapitulate, for example, genetic mutations that are strongly associated with these disorders. And this is what we've been doing in the lab for a few years, actually. And here, of course, you have two outcomes. Either we have like unifying signatures of autism, right, across all the the etiologies, or perhaps you have like different ones that you may or may not be able to cluster. And the result we got here, I think, are quite interesting, because first of all, what we show is that seemingly unrelated mutation can converge to impair the activity of certain circuits. Blue here means reduced connectivity in cortical areas, and as you can see, these four mutations produce reduced connectivity in prefrontal regions. However, if we zoom out, we begin to include more mutations, the spectrum is kicking in, so to speak. So you see that in this case, for these two mutations, we have more unimodal reduction in connectivity. We also have mutations like neuroligin when there is no cortical effect. And the last bit, which is the one I like the most, so to speak, it's, you really have the opposite effect as well. We have mutations that produce over-connectivity, right? So here is the first approximation of the spectrum, and the take-home message, and I think it's very clear here and very colorful as well, is that, genetic is indeed a key contributor, and we have to keep that in mind when we want to, to make sense of this heterogeneity. Of course, this is a first approximation of the spectrum, very few mutations, so we want to go bigger. So what we did, we teamed up with the super talented Valerio Zerbi and Nikki Vanderhout, ATH. I'm sure you've seen the poster on Monday. The idea is to join together all the mutations we have, because those guys are pretty much the same program. And long story short is that we pretty much have the same result. We have a hyper-connected cluster here. We have hypo-connectivity, 3 clusters here. And as mentioned before, there are 2 clusters, for example, that are anti-correlated. So far, so good. Now, let me just carry this forward and address the second question. Can we leverage functional connectivity heterogeneity to identify autism subtypes? Well, this is an attempt we did, and I'm going to show it to you. There was also a poster a couple of days ago, but I tried to sum up very quickly. And this was prompted by this study from David Salzer at Columbia. I'm sure many of you saw it a few years ago. Basically, what the Salzer lab demonstrated is, first of all, that in post-mortem autism brain, there's an excess of synaptic spines. And most importantly, in many of these spines, right, that these guys counted, they found that there is a specific biochemical signature. And this biochemical signature is called mTOR pathway hyperactivity. Now, I won't tell you, it's not important you guys understand what this pathway is. I'm sure many of you know what it is. But the key message is that this is a pathway that is very central to autism because many mutations converge onto it, like P10, TSC2, and so on and so forth. So the question we ask is, okay, this is a prominent pattern in autism, okay? Many kids have it post-mortem, right? So what is the functional connectivity signature associated with this hyperactivity of mTOR? So this is something that can be modeled very easily in the mouse, so we did it. And here, first of all, we have like a sanity check. We see that these mice have increased synaptic density, as you would expect. And when you look at the connectivity, we have overconnectivity, mostly in polymodal areas, striatum, polymodal, like insular areas, okay? What is interesting, though, is that in the mouse, you can also normalize this mTOR activity, for example, pharmacologically. And when we do that, we normalize both spine density and connectivity, which is then establishing a linking between these two phenomena. And so far, so good, so it's quite exciting. There's another interesting thing that we found that these changes are behaviorally relevant because we found that corticostriatal connectivity is in fact associated with autism-like stereotypies in the mouse, so it's another sanity check if you like. It makes all sense. But what about humans? Now, let me very briefly recap the story so far. So, David Salter said, post-mortem, a lot of spines, and many of these spines are associated with this mTOR hyperactivity. So this is a prominent effect in humans. And so we said, okay, there must be some signature of this hyperconnectivity that we found in the mouse, because we demonstrated that this signature means hyperconnectivity. We looked for it, we used degree weighted centrality, and we found two foci in insular regions, right? And this is a BIDE1, basically, in kids and in children, a BIDE1. And what is really interesting and encouraging is that when we map the connectivity between the insula and all the rest of the brain, we have a hyperconnectivity pattern that is very much similar to what we found in the mouse. We found that this hyperconnectivity pattern is significantly correlated with repetitive behavior in children. And last but not least, and very important, we found this pattern to be spatially correlated with expression pattern of mTOR gene interactome, which would suggest that in humans, this subtype of hyperconnected brain can be not exclusively, but probably prominently driven by this function. So I got 2 minutes to address the last point. I think I cannot make it because it's a very short story. The last story is, as mentioned, one question that's been there for a long time is that, it'd be interesting to understand how neural dynamics or alteration in neural dynamics could be related to functional disconnectivity in autism. And the approach we develop, again, in the mouse is an approach that is, by the way, of course, applicable also to humans, and we're doing that, is an approach whereby you can decompose resting state time series into states. These states are oscillating, so they are just reconfiguring, not in an abrupt way, but just they have like these infra-slow oscillations. And these states here can be mapped with voxel resolution, both at the subject and at the group level. So what we see here, blue is like a down in bold activity, red is an up. So you have ups and down on bold activity. You don't have correlation anymore, which is something that probably everyone likes here in the room, right? To just get rid of correlation for a while. And why do we like this? We like this because then we can use these measurements to understand whether dynamics in models of autism, so mouse that are models that are recapitulating changes, genetic changes associated with autism, can be mapped regionally. And what we found here is that we have atypical state topography. What does this mean? This means that these states that are recurring, that are very reproducible in controlled mice, in mutants, which is here the red up and down you can see here, they have non-canonical patterns of coactivation. It means that in certain specific areas, not throughout the brain, but in certain specific areas, these dynamics is disrupted, right? And so here we have one way to pinpoint where these regions are actually anatomically, and as mentioned, this is not a global effect, this is regional. And so my last slide here is, okay, so how can we somewhat relate these regional neural dynamics to functional connectivity, the static connectivity we use? Well, the answer, we believe, it's yes, or at least that we have preliminary evidence that this could be the case, because if you just regress out to remove a single state, for example, state 1 here, or state 2, you can completely block differences or remove differences in resting state connectivity, measuring in the traditional way, like static. So we believe this study here and this approach here is somewhat highlighting a novel phenomenon, which is the fact that this functional disconnectivity can emerge as a result of this underlying disrupted neural dynamics. So my time is up, but I want just to give you very briefly some take-home messages. First and foremost, I hope I have convinced you that there is scope for using cross-species fMRI to try and understand what happens in the clinic, to inform, mechanistically what we get. Keep in mind that etiological variability, genetics mostly, is a key contributor, to autism connection heterogeneity. We propose that that hyperconnectivity may be partly explained by hyperactivity mTOR pathway, and that's this new phenomenon, this aberrant oscillatory network dynamics that may be involved in also this functional decoupling we see in connectivity. We haven't addressed, of course, all the questions, but perhaps this is really a tiny ship of order here. So I have to thank all my funding agencies, especially the Simons Foundation, for generously funding this research, and of course our key collaborators, Marco, Stefano Panzeri and Danielle, and thank you for your attention.